this is James MSCC's final seminar, and thanks everyone for coming here. So James got his bachelor's degree in biology at Anderson University in May 2018, and then in August 2018, he joined us for studying his master's degree in hot science. So today is his final seminar. Thank you everyone for coming.
Yes, thank you. Okay, so I'll begin. So as he said, my name is James Duduy, and this is my master's final seminar for my thesis project on the coordinated transcriptional regulation of the carotenoid biosynthetic gene pathway genes which contribute to fruit lycopene content in high lycopene tomato breeding cultivars. So tomatoes are one of the most economically important crops in the entire world. And the US is actually the second largest producer of tomatoes following China. And in 2017, there was $1.67 billion of value produced from the US market alone. Originally, these fruits were domesticated in South and then Central America, but today the fruit's grown all over the world. So today, as I said, what we're interested in is lycopene. It's a secondary metabolite that's produced by tomato fruits, but it's also produced by many other fruits, including watermelons, strawberries, and then many of the others that are over on the right. So the value of tomatoes is primarily based on color as one of the primary factors. And so it's a key factor when considering consumer preference for fresh or processed tomatoes. And the majority of lycopene is based on, the majority of lycopene in Americans' diets comes from tomatoes. And this is mostly in the form of processed like sauces, like pizza sauce, pasta sauce, etc. So in the fruit, it has a photoprotective function, and this protects both the fruit and seeds from UV damage. And there's actually research that's been shown that shows that lycopene is the most powerful of all the carotenoids at quenching singlet oxygen from UV damage. So it's a powerful antioxidant in tomatoes, and this is due to the high number of conjugated dienes within its structure. And so in humans, it acts more as a dietary antioxidant. And this has been shown to reduce the risks of diabetes, cardiovascular diseases, and even cancer. So here I'll introduce the carotenoid biosynthetic pathway, of which we have lycopene here in the middle. Then up towards the top, we start with the MEP pathway, which is the methylerythritol phosphate pathway. And from there, we have our primary metabolites of isoprenoids. So isoprenoids enter the carotenoid biosynthetic pathway as precursors to all carotenoids. So then we begin the pathway with geranyl pyrophosphate 1, 2, 3, and then several others that are still being researched. Then we continue on to phytoene synthase 1, 2, and 3, which this is the first committed step in the pathway, one of the most important genes. Then we continue on to phytoene desaturase, zeta isomerase, zeta desaturase, finally carotenoid isomerase to produce lycopene. So all of these genes up here represent the upstream because to us they are upstream of lycopene. Then we begin the downstream pathway, which branches off into two different paths. So we have beta carotene on one path, and then if we continue on down that way, eventually we get to neoxanthin, which then can lead to produce or synthesize ABA. If we go on the other pathway, we start with epsilon lycopene cyclase, which goes down eventually to produce lutein. And so the general way that this pathway functions is as we start upstream and then move towards lycopene, we are producing more of it. And then once we begin to metabolize lycopene, we begin to break it down, and so of course we get less of it. So some of the most important carotenoids that we care about in this study was phytofluene, which is an important metabolic colorless precursor to lycopene. And it's produced by phytoene desaturase, which again represents an important rate-limiting step in the pathway. Then after that, of course, we have lycopene, which I've already introduced, but it's a red carotenoid pigment. It's produced by many fruits, has a photoprotective function. And after that, post-lycopene downstream, we have beta carotene. which is the second most sequestered carotenoid in most tomato cultivars after lycopene. And this one is produced by two reactions with beta-lycopene cyclase 1 or 2. So the carotenoid biosynthetic pathway is mainly genetically controlled. Again, we start at the top of the pathway with our isoprenoid precursors, and then as we go down, we begin to see that many of the genes take different roles in different areas or different tissues. So lycopene and all other carotenoids are produced in plastids, and I'll get more into detail about that later, but the plastids where lycopene is sequestered begin as photosynthetic tissue, which are characterized by these green arrows trying to show, and then later the plastids where lycopene is produced become non-photosynthetic. So there's also many environmental factors that can influence carotenoid development. So these include, but are not limited to, increased temperature and light intensity. So now I'll go over some of the most important mutations that are known to be in tomatoes. So first we have yellow flesh, which is a PSY1 mutation, or phytoene synthase 1 mutation. So it's a loss of function of PSY1. So again, this is the very first committed step in the pathway, and it's also a really important rate limiting step. So this gene, when it loses its function, we see that we have a yellow flesh on the tomato because we're not able to produce the lycopene as would be expected. And so this gene actually is very important in the pathway, and we'll talk about it more later, but it's the highest expressed at both early stage tomatoes and late stage. Another mutation is tangerine, which is a mutation of carotenoid isomerase. And so we can see that here in the middle. This is the gene that actually is involved in the synthesis of lycopene. And so here we have another loss of function mutation. This affects carotenoid isomerase to effectively block any significant biosynthesis of lycopene. So this ends up leading to a high sequestration of pro-lycopene, causing the tomato to take on an orangish color. Next, we have old gold, which is a mutation of beta-lycopene cyclase 2. And so this one is actually a promoter mutation. And so this leads to an accumulation. So we're, in this case, we actually see an increase of lycopene at the expense of downstream metabolites, especially beta carotene, which is why we see a much darker red color on the tomato. So these were all pathway mutations that I've just been talking about, but there are other mutations that can increase lycopene that are not related to the pathway. And so I'll go over those right now. So first, carotenoids are produced and sequestered in chromoplasts in vivo. So they start as chloroplasts, but then eventually convert to chromoplasts as the tomato develops. And this is for the purpose of carotenoid sequestration in tomatoes. So some mutants that can take advantage of this are high pigment or HP mutants, and these are characterized by their larger and or more numerous chromoplasts. So here we see HP1, which is UV damage binding protein 1 mutation. We also have HP2, which is deethylated 1 mutation, or DET1. And so from these, we can see increased size and number of chloroplasts as well as chromoplasts. So the image I'm showing here shows HP2 mutant on top with its wild type below it. So we can see that we have chloroplasts here, so immature chromoplasts, where we see much higher concentration as well as larger size of these chloroplasts in relation to its wild type. And we can also see this with the ripe fruit as well. So here we see on the top, if we look at the flesh of the tomato, much, much deeper, darker red than its wild type counterpart. So there's mutations such as old gold or high pigment that are used in agriculture in order to significantly increase the amount of lycopene produced. But typically these mutations tend to have a little bit of problems. So they end up being crossed a lot with many different genetic backgrounds. And so that's how we have so many of our high lycopene cultivars. So many past studies have focused on low lycopene genes that have been genetic studies. So they've been relatively small scale. So first, I'll just say that a low lycopene line is defined as being 0 to 40 micrograms of lycopene per gram of fruit, fresh weight, and that's a low lycopene line. We'll talk more a little bit about that later. But so these studies that have been genetic tend to be relatively small scale, few lines, 1 or 2 stages, only a few pathway genes. And then they also tend to only focus on upstream genes, especially PSY1 and Z-ISO, which are very important genes. And then they've also, of course, found a general trend, as expected, of high expression in the upstream genes and then low expression in the downstream. So there's also been some studies with high lycopene lines. However, these tend to not be genetic in nature. They're more field studies of agronomic performance. They focus on carotenoid content, fruit yield, size of the fruit, and then also how crossing with different genetic backgrounds may cause different agronomic performance in the field. So why would we want to do this? Well, there's not been any previous genetic research in so many of these high lycopene lines. And so this is the first genetic study to see why all of these diverse high lycopene breeding lines produce high lycopene. And so again, first study to look into whether and how transcriptional regulation of the pathway genes contribute to the high lycopene fruit content in all of these lines. So the goal that we took was to use 42 high lycopene breeding lines from around the world, along with 1 wild tomato relative, 1 heirloom tomato, and then 1 non-red tomato, in order to correlate biosynthetic gene expression along with carotenoid fruit content, especially lycopene, but others as well. So this information should later on down the line allow for further improvement of lycopene content via genetic engineering, gene editing, or breeding. So here's a list of all of the lines that we used in this study. And I know it's a lot to take in, but it's really the point that we're trying to get across here is that we have many, many diverse lines with different mutations, mostly unknown mutations. But these lines today come from international and domestic tomato breeding companies. So we have lines from United States, Italy, France, Japan, Spain, Israel. So many, many diverse domestic and international lines that have been developed specifically to be high lycopene. So our tomatoes were all grown in a Kannapolis greenhouse at the same time from August 2018 until March 2019. We kept the temperature at a normal greenhouse temperature, 3 biological replicates kept in 3 separate pots for the 45 lines, and then these were all in randomized complete block design throughout the greenhouse. So you might be wondering how we defined the different stages of tomatoes. So we had to collect so that we could look at the different stages as the tomato developed to see how the gene expression changed as the amount of carotenoids increased. So one of our earliest stages, breaker, the earliest that we actually used is breaker. And so this one is defined as being, as showing a break in color from the light to dark green that we see from the earlier stage tomatoes. Then after that, we have orange, which is where 30 to 60% of the surface shows an orangish hue on the fruit. Then we see pink, which is where 60 to 70% of the surface shows a pink to light red color. And then finally ripe, which is where 90% or more of the surface shows a deep or strong red coloration. So it's actually easier to see and determine what stage is what if you're actually in the greenhouse. So whenever you're actually looking at the plant and you can compare stage by stage, it's much easier. So we see here at the bottom, we have immature green, then above that, which immature green is defined as being not its full size yet. Whereas we have mature green right above it, which is defined as being the full size that it will be. Then we have breaker, which is full size, but then begins to show a break in color. And then of course, orange, pink, and ripe. So these fruits were collected and sampled based on their maturity stage. Again, 45 unique lines, very large scale project relative to what's been done before. And then we took 4 fruit developmental stages for HPLC, which were the breaker, orange, pink, and ripe that I showed you. And Dr. Penelope Perkins actually did all of this HPLC work for us, so we owe a lot to her. And then we used 2 fruit developmental stages for RT-PCR, which are breaker and ripe. And then Paul Kosenka and I both split the work of that half and half. So we moving towards qPCR, RT-PCR that we did, we had to design internal reference genes because these internal reference genes are the baseline for when we're assaying all of our genes throughout the biosynthetic pathway. So typically these internal reference genes are housekeeping genes which have relatively stable expression across tissues and different experimental treatments. So our design, we included 2 internal reference genes, these being clathrin adapter complex, or CAC, and then expressed, or EXP. And then, so we selected these 2 internal reference genes based on their high reference stability and then how they function together when assaying tomato fruit or tomato tissue. So looking at these genes and comparing them together based on the data from the tomato expression atlas, we can see that, okay, so we have our different stages going across the top from breaker, pink, and red at the end. So then we see down here, we have total pericarp, which is the tissue that we care about, but I'll talk more about that later. And what we're looking for here is to have a relatively similar expression all the way across, so that whenever we are assaying all of our different genes, that we can actually compare them equally. And we see the same down here with EXP. So we had to design primers in order to perform qPCR. So we performed a single nucleotide polymorphism-based primer design. So these are primers that are designed uniquely around SNPs so that we have homologous sequences that can be distinguished. So we wanted a high degree of specificity across our different homologous sequences so that when we amplified, our CyberGreen only gives a single amplicon. And then finally, we designed 4 primer pairs per gene so that during the optimization steps, we could have a higher quality optimization. So we have our protein sequences that we originally received from all of the biosynthetic genes, and we took that to GenBank and used it to query search the entire tomato genome. So most of these genes are single copied, but some of them have more. So we had to look and remove the ones that lacked apparent sequence homology, and then that left us with a total of 25 biosynthetic genes, which are highlighted over here, and then 2 reference genes that were found in the phytosome. So the protein sequence alignment for the 25 genes, we found that they could be categorized into different groups. So we designed a phylogenetic tree, seen here, which is used to design primers, and this is all based on the similarities of the sequences. So we ended up observing that there are 12 different groups that have very high sequence homology. So these were all compared to each other when we were designing primers. So the next step after that was we took the cDNA sequences and then aligned it in a clustol X within each of those 12 groups. And here we included the untranslated regions, and then we preferred our primer design towards the 3' end. So we designed, as I said earlier, we designed 4 primers, 2 forward, 2 reverse, and these were designed close to each other for all 27 genes. So we can see over here we have PSY1, 2, and 3 that are all aligned to each other. So when looking at our 2 forward primers, we have our forward 1 that is underlined and our forward 2 that is highlighted. And so when designing our ends, our 3' end for our forward primers, we preferred to have either a C or a G. And this was in order to have higher binding ability as well as higher specificity across the different homologous sequences. So you can see we preferred a C or a G, as well as having these end nucleotides to be different from the other homologous sequences. And you can see that also for the 2 reverse primers, where we have but it's just in the opposite direction in the 5' direction. So we had specific design limitations that we had to abide by, these being 20 to 23 base pairs in length per primer, 40 to 50% GC content per primer, and then 85 to 125 base pair amplicon was ideal, and this with the 125 including the primers, both forward and reverse. So then we had to optimize our primers. The first step being annealing temperature optimization, which is where we have a temperature gradient seen here, and the goal being to find the lowest CT value. And then following that, we did primer concentration optimization, which is where after we found our ideal annealing temperatures, then we performed primer concentration with a gradient of different primer concentrations in order to find the lowest CT value. So this is all a setup for serial dilution, which is the main point of doing all of this. So the goal is to produce a standard curve, which we can see right here, and this will inform the acceptable dilution amount for the cDNA concentration range. So all of this being the goal to find an R-squared value higher than 0.99, and then an efficiency of about 100 to 105%. And so we wanted to find, so the goal was to find the best primer pair for each gene. And we had to do this in the end in order to do the delta-delta CT method, which is one of the most common and important data analysis tools for qPCR. So the tissue that we actually used in our sampling is called pericarp, which is sort of the outermost part of the tomato. So this includes the outer skin out here, and then it also includes the inner flesh, all the way to the inner pericarp here. So we sampled our tissue by cutting it away, and then we stored it in -80 for later carotenoid HPLC sampling. And then in the case of RNA extraction, we had to freeze it in liquid nitrogen and grind it to a very fine powder. And the reason why we use pericarp is because pericarp has very equal expression throughout the tissue type. And we wanted to keep to only one tissue type, because if we were to mix our different tissues, you'd see that the expression is not equal throughout all tissues in the tomato for all of our genes. And so in order to keep it as stable and expected as possible, we kept to only pericarp. It's also very common within literature that only pericarp is used. So the carotenoids that we looked at via HPLC were phytofluene, which represents the upstream part of the pathway, or pre-lycopene. Then we have lycopene, which is the most important one that we care about. And then we have our downstream beta-carotene. So we also did DIC microscopy for imaging. So with DIC microscopy, or differential interface contrast, we're actually able to optically dissect a single layer of cells. So it's essentially a light microscopy in order to discern plastid number and size versus the cell size. So these individual cells can be imaged and counted. And actually here we have images of some of our ripe stage tomatoes and their cells. And if you see within some of these red dots, these are the plastids that we care about, the chromoplasts. So here's an image of all of our ripe stage tomatoes that were imaged. So again, we have 45 lines, 42 of which being high lycopene, but I'll point out the ones that are not high lycopene lines. So for example, we have number 1, which has the most amount of lycopene. This is our wild tomato relative. It's Solanum pimpinellifolium. Then down here, number 44, we have Moneymaker, which is a commonly grown heirloom. It's been grown in greenhouses for 50 or more years. And then we have Tangerine right here, or sorry, NC1Y, which has a tangerine mutation. So this is the mutation of carotenoid isomerase, which is directly involved with producing lycopene. So this one has a much higher concentration of prolycopene. So now we'll talk about some of our results. So down here on the x-axis, we have all of our tomato lines. We're just looking at lycopene in this graph here. So we have all of our lines listed from highest amount of lycopene, which is our wild tomato, down to the lowest amount at the far right with Moneymaker, and then NC1Y, which had no lycopene. So we can see that our y-axis is fruit lycopene content of micrograms of lycopene per gram of tomato fresh weight. So also note here that our graph only goes up to 350, so that will come back up later. So yeah, I don't know if you can see this, but at the bottom we have our different stages of tomatoes. So the red color being ripe, the blue color being pink stage, orange color being the orange stage, and then green color being breaker stage of tomato. So Moneymaker is one of our controls that we used in this study. So we intentionally want to compare all of our other lines against it. So in doing that, if we look at our two highest amount, or our two lines that had the highest amount of lycopene at the ripe stage, we see that our wild tomato had 5 times the amount of lycopene as Moneymaker. And then our next highest, high lycopene line from France, I believe, ISI 12152, that has 3 times the amount of lycopene. So then we can also do the same thing with beta carotene. We have beta carotene content by micrograms of beta carotene per gram of tomato, fresh weight. So once again, we have all of our lines in order from highest amount of lycopene at the ripe stage down to the ones that had the least amount. So here you'll already notice that our graph is much lower in this case. It only goes to 50. And there's not as clear of a pattern that can be discerned from this. It appears to be much more random and much more varied across the different lines. We see a similar sort of result with phytofluene, where phytofluene is even less concentrated in each of the tomatoes. So we see just significant variation across the different lines, and a where we can't really discern any sort of pattern from this. So now I'll show you how the different carotenoids actually increased as the tomatoes developed. So first, our colors change here a little bit. We have breaker is blue, orange is orange stage, green is pink stage, and ripe is red stage. So it increases. So here we see that most of the lines actually increase from stage to stage, where we see our highest amount of lycopene tomato, HLY18 in this case, it increases from orange to pink to ripe. So you might notice that we are missing some of our stages here. It's because we don't have full data sets for all of our tomatoes. So in this case, I'm only showing the tomatoes that we have full data for. And so we see that most of the lines going across increase at an expected rate, but we see that also, for example, LYCO2 and then HLY13 down here at the right do not increase at a normal rate, but actually pretty much maximize their amount of lycopene at the pink stage. Now, not much can be gleaned from the beta carotene actually in this case, where from stage to stage, we see some of the lines actually increase at a normal rate from increasing from breaker to orange to pink to ripe. However, there are some, especially Crispino F1 and Crispino Plum, which almost maximize their amount of lycopene at the breaker stage. So again, not much can be gleaned from beta carotene. So here we see fruit lycopene content, and again, there's not much of a pattern here, except for many of the lines have a more normal rate of increase as lycopene did, more so than beta carotene. However, some of these like HM5235 here do not. So now we're talking about qPCR results. So this is a very large heat map that's going to be a little bit difficult to unpack, so I'll take it slow. But we have all of our lines over here on the left. So we have wild tomato at the top, so our highest amount of lycopene at the ripe stage tomatoes up here at the very top. And then going down, we have less and less until we finally get to Moneymaker and NC1Y, which had no lycopene. Across the top, we have GGPPS1, which is the top upstream portion of our pathway. And as we go to the right, of the pathway, we get to carotenoid isomerase, which is the gene that's involved in the production of lycopene. So our scale here is where red indicates higher expression. And then as we go down, we get to white, and then we go down to blue, which indicates very low expression. So first, I want to, one thing first is that we have 2 boxes here for each of our genes. So the box on the left for each gene indicates breaker stage tomato data, and then the box on the right indicates ripe stage data. So that way we're able to compare each stage to each stage across all of our tomatoes and across all of our genes. So one thing that I want to point out first is that for NC1Y, our tangerine mutation tomato, it has the expected result that we were expecting. So we see that carotenoid isomerase, whereas for every other tomato is rather highly expressed, for NC1Y, it has very low expression. This is as expected because this tomato only produces high amounts of prolycopene and not lycopene. However, across the rest of the upstream portion of the heat map, we see that going down for each gene, most of these lines have relatively similar expression. And so this is an interesting pattern that we didn't fully expect. But we also see that with the downstream portion. So we're starting the downstream portion. We have beta-lycopene cyclase 1 and 2 up here at the top, going down to NSY1, which ends the right portion of the pathway, of epsilon lycopene cyclase, which starts the next portion of the pathway down to CYP97C11. So again, you'll see that going down each of the genes, that there's relatively similar expression across the genes and across all of the different lines. However, if you look at the general trend of the downstream pathway compared to the upstream, you'll notice that the upstream actually has much higher expression overall than does the downstream, especially for genes that initiate the pathway. So we have GGPPS2 and 3, and then PSY1, which initiate the upstream pathway, whereas we have beta-lycopene cyclase 1 and 2 and epsilon lycopene cyclase that initiate the downstream pathway. So these are much lower in expression than the upstream pathway, which just goes to indicate how most tomatoes do have this general trend of high expression upstream and then low expression downstream. So now here we compared all of our 42 high lycopene lines against NC1Y mutant carotenoid isomerase gene. So on the top we have bar graphs where our green bars indicate NC1Y carotenoid isomerase, and then our blue bars indicate the 42 lines grouped. So on the top we have our breaker stage tomatoes for all of our genes, and then on the bottom we have the same, but just for ripe stage. So once again, our genes are all in order in the same way as the pathway. So starting upstream with GGPPS1, 2, and 3, and going down to carotenoid isomerase, which produces lycopene, then initiating our downstream pathway, beta-lycopene cyclase 1 and 2, and then finally ending it. So, and then also these are our relative expression values for all of these genes, all of these tomatoes in a log scale. So now I'll highlight all of the genes that are insignificant, or that are insignificantly different from this mutant NC1Y carotenoid isomerase. So the genes that I've highlighted being TPT2, PSY3, beta-lycopene cyclase 1 and 2, BCH2, and then NSY and epsilon lycopene cyclase, all of these are insignificantly different from the NC1Y mutant carotenoid isomerase. So what this indicates is that all of these genes are either silenced or they have very low expression, because they're very similar to this mutant carotenoid isomerase gene. So these are genes that are not very important to the overall flow of the pathway. And then as we move on, again we have the same order of genes, but this time we're comparing Moneymaker, which is our green bar, to our 42 high lycopene lines grouped. So Moneymaker, just to remind everyone, is our red tomato that had the lowest amount of lycopene content. And so here we're looking at all of the genes that are significantly different, not that are significantly the same, but significantly different. So I've highlighted those here. So most or all of these genes are more highly expressed in the 42 lines grouped. So we have 8 upstream genes and 8 downstream genes that are significantly different from from moneymaker's expression of those genes. So this is very significant because this indicates that there's a higher metabolic flux flow from the upstream MEP pathway into this pathway, all the way to the downstream portion of the lycopene biosynthetic pathway. And now here we're comparing wild tomato, which is the green bar, against our 42 high lycopene lines grouped, which is the blue again. And then here again we see that we have many significant genes, but more so in the downstream portion of the pathway. So remember that wild tomato had the highest amount of lycopene at the ripe stage as compared to any of the other tomatoes. And so these upstream genes probably don't matter as much for wild tomato, but the downstream genes do appear to consume more of the lycopene in the 42 high lycopene lines as compared to the wild tomato. So now here's just a general overview of the pathway where we can look at and see along the pathway which have the insignificant difference, the breaker stage or at the ripe stage, when we compare our 42 high lycopene lines to our NC1Y mutant carotenoid isomerase. So here's where they fall. We have, we have TPT2, we have PSY3, epsilon lycopene cyclase, beta lycopene cyclase 1 and 2, BCH2, finally NSY, that are all insignificantly different. So, sorry, again, those are either silent or very low in expression. So now here we can look at the pathway as it relates to moneymaker. So the up arrows represent significantly higher expression at either blue for breaker stage or red for ripe stage. And we have a few genes that are lower in expression. And again, same color, just pointed down for lower expression. So we see that mostly our downstream genes have significant differences. So just overall, we're able to quickly see which genes have significant difference and which might contribute to the higher amounts of lycopene within our 42 high lycopene lines. So going back to chromoplast analysis, it was actually very difficult to measure the plastids of the ripe stage. We could see the size and the number of the size and number difference of the chromoplasts, but image analysis was not really possible at this stage. So you can see that there's many chromoplasts in here. They ended up being very, very difficult to image or to count with any imaging software. So we did start with ripe stage looking into this, and we did find this unique number and size between lines, but we weren't able to count it. So we played with several different stages, and eventually we found that mature green is much better to measure and to count. So we found that the chloroplast of mature green stage, much easier, and here you can actually see that that we were able to find a consistent relationship between the size of the cell and the area of the plastid. So you might be wondering if mature green is representative of red or consistent. Yes, as the fruit matures, the amount and the size of the plastids does stay normal to the size of the cell overall as it expands. So there is a relationship here, but it's just that with mature green, it's much easier to count than it was with ripe stage. So we still have more work to do on that. But our overall take-home messages are that we found a general trend of strong expression in the upstream genes. This was prior to lycopene biosynthesis, weak expression in most downstream genes at both stages. We see a clear increased expression of our grouped upstream and downstream genes compared to moneymaker and the wild tomato. Our upstream genes listed on our top bullet point, and then our downstream genes listed on the bottom bullet point. And our data suggests that coordinated transcriptional regulation of these pathway genes can lead to a higher metabolic flux flow of the pathway, which contributes to higher lycopene content overall. And we also may be able to see that chromoplast data can reveal new factors, maybe through chromoplast size or number, as it contributes to high lycopene content, but we still have more to do with this. So I would first like to thank my committee in helping me with this, especially Dr. Wusheng Liu, who helped develop this project. He supported Paul and I throughout this entire process, and he also helped guide me as I learned all of the different techniques and procedures. I'd like to thank Dr. Penelope Perkins for supporting this project by providing greenhouse space for the tomatoes, as well as for HPLC analysis, which is critical to this project's goal. And she also performed HPLC on some samples that were not included in this project, including immature green stage and mature green. So I would also like to thank Paul Kosenko, who split the qPCR work with me half and half. And he also imaged all of those tomato lines that we saw. And he's also been working on the chromoclast microscopy. And I would also like to thank Dr. Dilip Panthy, who provided seeds for project and guidance. And as well as many of the names listed here, Morgan Miller, Debao Huang, Liwei Gao, Shenlian Chen, Yonghui Wu, and Caitlin Dudui, who all helped so much with either tomato collection, grinding, or RNA extraction, or all three of those. So they have all been such a great help throughout this entire project. Thank you.